of biology, BioLessons to Go. In this video, we'll be exploring Mendel and the patterns of inheritance for AP Biology. Gregor Mendel was an Austrian monk who discovered the basic principles of heredity through experiments with garden peas. Mendel's observations became the foundation of modern genetics and the study of heredity. For this reason, he is widely considered to be the father of genetics. Even though farmers had known for centuries that crossbreeding of animals and plants could favor certain desirable traits, Mendel's pea plant experiments conducted between 1856 and 1863 established many of the rules of heredity and are now referred to as the laws of Mendelian inheritance. Pea plants make for excellent specimens for studying genetics. For one, you can control their mating very easily. By allowing pea plants to self-pollinate, it allows us to create purebred varieties. Or, if we manage their breeding and cross-pollinate the pea plants, we can create unique hybrids. Pea plants have many easily distinguished characteristics which we can track from generation to generation, including seed shape or seed color, flower position, or plant height. And finally, it doesn't take long for a pea plant to germinate and reproduce, so we're able to create multiple generations in a very short period of time. In a typical experiment, Mendel would begin with a parental generation, also known as his pea generation. This initial generation would consist of two contrasting true breeding varieties, where we would cross two purebred individuals in a process called hybridization. In this particular example, we'll be crossing a purebred purple flower with a purebred white flower. The resulting offspring are called the F1 generation, or the first filial generation, where filial is Latin for sons. This F1 generation, all of the resulting offspring are purple. Mendel reasoned that the reason for this is that only the purple flower factor was affecting the flower color in these F1 hybrids. He observed similar effects with all of the other 7 pea plant traits that he examined. One form appeared to be dominant over the other, which is to say that it masked the presence of the other trait. So in this case, the purple trait must be dominant over the white trait. He then allowed his F1 individuals to either self-pollinate or to cross-pollinate with other F1 individuals to create his F2 or second filial generation. In this particular example, we end up with about 75% or 3/4 of our offspring showing the dominant purple characteristic, and about 1/4 or 25% showing our recessive characteristic. Mendel assumed that it's likely then that the dominant inheritable factor didn't alter the recessive inheritable factor in any way. Both alleles could be passed on to the next generation unchanged. As a result of these experiments, Mendel came to several conclusions. He determined that the inheritance of each trait is probably caused by some inheritable factor that's passed on from generation to generation. Each individual seems to inherit one of these inheritable factors from each parent for each trait. And though a trait might not show up in an individual, it can still be passed on to the next generation. Today, we call these inheritable factors genes, and the different varieties of these genes we refer to as alleles. The alleles are carried on chromosomes, which are passed from parent to offspring as a result of meiosis and sexual reproduction. In this example, we're looking at a pair of homologous chromosomes from our F1 pea plants. Because they received one allele for purple from one parent and one allele from white from the other parent, they're still able to appear purple because purple is dominant. We would represent that dominant allele with a capital letter, like a capital letter P, and we'll represent our recessive allele with a lowercase p. We can then explore Mendel's experiments using this knowledge of genes. Our purple flower will have only genes for purple, so we'll have 2 dominant purple alleles. Our white flowers will only have the recessive alleles for white. Since each of these varieties have identical forms or alleles of the genes for their traits, we say that these individuals are homozygous. Our purple plant is homozygous dominant, and our white plant is homozygous recessive. When we describe their genes, we're describing their genotype. When we describe their physical appearance, we're describing their phenotype. When our P generation reproduces, they'll pass one of those inheritable factors, one allele from each will be passed to their offspring. So the resultant F1 generation will get a dominant purple allele from their purple parent and a recessive white allele from their white parent. Because they're receiving one of each allele variety, we say that they are heterozygous for this trait. They're phenotypically purple, but their genotype is heterozygous. When the F1s reproduce, some of the F2 purples can receive dominant genes, and those would be homozygous for their trait, while others will receive one dominant and one recessive. The white offspring will only receive recessive alleles. The only way to express our recessive phenotype is to be homozygous recessive. Now, through his experiments with pea plants, Mendel discovered several things which he concluded in his principles or laws. One such law is the law of segregation, which states that the two alleles for each character are not going to blend together in their F1 generation, but they stay separate inheritable units that can separate during gamete formation. So our purple flowers, which have one purple allele and one white allele, those alleles didn't blend to form some new inheritable factor. Instead, the purple factor, the purple gene, stayed separate and was able to separate away from our white allele in the formation of those gametes. Another major principle or law that Mendel was able to determine is his law of independent assortment, which states that when two or more characteristics are inherited at the same time, each pair of alleles will segregate independently of each other during gamete formation, allowing for equal opportunity of those occurring together. So in this particular, the yellow characteristic being inherited with the wrinkled, or the yellow characteristic can be inherited with the round factor. In the same regards, the green characteristic can be inherited with the round factor, or the green characteristic can be inherited with the wrinkled factor. If these factors could not separate independently of each other, we would always see the yellow going with the round and the green going with the wrinkled. But because they are able to create separate varieties, it tells us that these alleles can separate independently of each other. This concept of independent assortment can be illustrated further when we re-examine meiosis. When those chromosomes align at the equatorial plate during metaphase 1, as long as the alleles are on separate chromosomes, they can align independently of each other, which allows for the recombination of those alleles in a number of different patterns. Now, much of what Mendel observed can actually be explained through mathematics and the laws of probability. For example, we can use the rule of multiplication to determine the chances that two independent events will actually occur together in a specific combination. The way that we do this is we multiply the probabilities of each independent event with each other. So, for example, if we were to throw two dice, what would be the chances of getting sixes both times? Well, the chances of getting a 6 one time with a dice is 1/6. The chances of getting two sixes right after each other is 1/6 times 1/6, which is 1/36. In terms of genetics, what is the chances of having 5 boys in a row? Well, the chance of having a boy is about 50%. The chance of having 5 boys in a row is that independent chance multiplied by the other independent chances of that event happening again. So it's 1/2 times 1/2 times 1/2 times 1/2 times 1/2, or 1/32. Another application of the rule of multiplication would be if we crossed an individual that was homozygous A, heterozygous B, and heterozygous C with an individual that was heterozygous for the A trait, heterozygous for the B trait, and heterozygous for the C trait, what's the chances of us having a particular offspring that's heterozygous A, heterozygous B, and homozygous recessive C? Well, in order for us to complete that, we have to determine the independent chances of each of these events taking place. And you can either do this in your head, or you can actually create some little Punnett squares to be able to test this out. So if we have the independent event of a homozygous A crossed with a heterozygous A, the chances of getting a heterozygous A is 50%. So we have a 1/2 chance of getting that. A heterozygous B crossed with a heterozygous B, we have a 50% chance of getting a heterozygous B. And then heterozygous C crossed with heterozygous C, we have a 25% chance of having that happening. So when we multiply 1/2 times 1/2 times 1/4, we're going to get 1/16. Another good law of probability that we can use is the rule of addition. The rule of addition is used to determine the probability that two mutually exclusive events will occur. When we need to figure this out, we're going to add together the individual probabilities. For example, if we were asking, what are the chances of throwing a dice that will land on a 4 or land on a 5? Here, it's 1/6 chance to get a 4 and 1/6 chance to get a 5. To get either or, it's 1/6 plus 1/6, which is going to be 1/3. In genetics, if we were to examine what's the fraction of the offspring from a heterozygous cross that will have either the dominant phenotype or the heterozygous phenotype. So when we look at our little Punnett square in our cross, there is a 1/4% chance, or 1 out of 4 chance, that we're going to be homozygous dominant, and there's a 1/2% chance of being heterozygous. So 1/4 plus 2/4 is going to equal 3/4, or 75%. Mendel's discoveries were just the tip of the iceberg. With the rediscovery of his work, scientists began to uncover other patterns of inheritance that we'll be able to observe very shortly.